the students. So the next part of chapter 3 is fertilization. So fertilization is, we all know, it is the fusion of the male and the female gamete. So previously in the previous videos you have learnt how the male gamete is formed, how the female gamete is formed. So the next step is fertilization, means after copulation the male gamete will enter the female reproductive tract and their fertilization will occur. So now we'll see the various steps of fertilization. So it is the fusion of male and female gamete to form a diploid zygote. Diploid means twice n number of chromosomes. Diploid zygote. And where does fertilization occur? Very important. It occurs in the ampulla region of fallopian tube. Fallopian tube is present in the female reproductive system. It occurs in ampulla fallopian tube when between 14th to 16th day of the menstrual cycle. Okay, this is when fertilization will occur. Means in 14th day the egg is formed, ovulation occurs. So between 14 to 16 day fertilization will occur when the sperm will meet the egg. Now the different steps of fertilization. First is approach of the sperm. Approach of sperm means the sperm will enter the female reproductive tract. The sperm should reach the egg. So that is approach. So in one ejaculation of male, about 3 to 3.5 ml of semen is deposited. Okay, so this in 3 to 3.5 ml of semen, there will be about 200 to 300 million sperms. Okay, in one ejaculate once, about 3 to 3.5 ml of semen will be deposited in the vagina. So from there 200 to 300 million sperms will be there. So these sperms, there will be some factors which will help the sperms. There will be also some barriers to the sperm. So after crossing the barriers, about 100 sperms are left. Only 100 sperms will reach the fallopian tube. This we'll see later. First we'll see what helps the sperms to reach. So what helps the path of sperms? First is lashing movement of the tail of sperm. Lashing movement of tail. The prostaglandins. Prostaglandins. These are responsible for movement of the sperm from vagina to upwards, up to the reproductive tract. Then aspiratory movement of uterus. Aspiratory movement of uterus means the sperms move from vagina. This is the aspiratory movement of uterus will help the sperm to move to the vagina. Now these three is what helps the sperms. There will be also barriers. Now we'll see what are the barriers. The thick endometrial lining of uterus. We know there is an endometrium in the uterus. So the thick endometrial lining of uterus will help the sperms to cross. Next is the thick endometrial lining will stop the sperms from crossing. Okay, because it is a barrier. The acidic pH of the female reproductive tract, this will kill the sperm. Thirdly, there will be certain phagocytes which will eat up the sperm cells. So after getting this help, crossing these barriers, there will be finally about 100 sperms which will reach the fallopian tube. The rest will die. Okay. So these sperms, they have a certain speed at which they move. They will move at the rate of 1 to 4 millimeter per minute. This is the rate at which the sperms move, 1 to 4 millimeter per minute. So finally, we have got 100 sperms. Now, this is the first step, approach of sperm. The next is, we'll write it as AB. Fertilizing, anti-fertilizing reaction. Fertilizing, anti-fertilizing reaction. Now what is fertilizing and anti-fertilizing? Fertilizing is secreted by ovum. So this fertilizing will attract the sperms. So the sperms will get attached to the ovum. So this fertilizing is secreted by ovum. Similarly, anti-fertilizing is secreted by the sperms. So when these two get attached to each other, means what? There will be identification. Means the sperm and the egg will identify each other. Identification of both sperm and egg. That is fertilizing, anti-fertilizing reaction. Now, why is this identification required? Firstly, to maintain the uniqueness of species. Maintain uniqueness of species. Secondly, we don't need so many sperms. Ultimately, we need one sperm. So, in order to thin out the number of sperms. And thirdly, so that few sperms bind to egg. Few sperms will bind to egg. Out of that, only one sperm will enter the egg. So, for these three reasons, there should be identification between the sperm and the egg. Okay. So, this is A and B. You can note down from here. Next, we'll see step number C. What is step number C? Release of sperm license. Identification is done. Now the sperms will release some lytic enzymes, which are known as sperm license. Okay. So these lytic enzymes are of again three types. One is corona penetrating enzyme. Second one is hyaluronidase. And the third one is acrosin. Now we'll see the functions of the three. We know there are certain membranes covering the egg cell. So the membranes should degenerate, or the membranes have to be gone so that the sperm can enter the egg membrane or enter the egg. That is why these three sperm licenses are required. So these sperm will release these enzymes and the membranes will be gone so that sperm can easily enter the egg. So this corona penetrating enzyme, as the name suggests, this will digest the corona radiata. Digest the cells of the corona radiata. Hyaluronidase, this will digest the hyaluronic acid. This is present, hyaluronic acid is present as a glue in the cells surrounding the egg. So this hyaluronidase will digest those cells. Then acrosin will digest zona pellucida. This will digest corona radiata, this will digest hyaluronic acid, and this will digest zona pellucida. So the membranes are now gone, sperm is free to enter. Now, the next step is formation of receptor cone. This is also known as fertilization cone. Now, suppose, remember this is the egg. There will be, when the sperm will come from this side, a certain cone-like conical structure will be formed here. This is the receptor cone or fertilization cone. This conical structure is present on the animal pole, because through animal pole sperm enters. This is present on the animal pole, sperm will attach to it, this membrane will be gone off and the sperm will enter the egg. Okay, so after this, the next step is the start of meiosis 2. Remember when we studied oogenesis, I told you meiosis 2 gets started only when the sperm comes in touch with the egg membrane. Sperm and egg membranes come in touch. So as soon as the sperm comes in touch with the egg membrane, meiosis 2 will start. So this meiosis 2 will convert the secondary oocyte to ovum and a second polar body. The second polar body will degenerate. We need this ovum. Okay, so after this we have got the ovum now. Next is cortical reaction will occur. Cortical reaction or formation of fertilization membrane means the egg has exoplasm here. This exoplasm has cortical granules. These are cortical granules. These cortical granules will go here. This space is known as perivitelline space. Perivitelline space. So this cortical granules will come to perivitelline space as soon as ovum is formed and attach to the inner surface of the vitelline membrane. Means it will attach here. Now what will this cortical granules or cortical reaction help? Because already one sperm has entered, we don't want any more sperm. So as soon as this cortical reaction occurs and the cortical granules form a layer in the perivitelline space or in the inner part of vitelline membrane, no more sperms can enter. No more sperms can enter. So this cortical reaction prevents polyspermy. Polyspermy means no fusion of more than one sperm. Next is fusion of gametic nuclei. This is the final step. Fusion of gametic nuclei. Now what is gametic nuclei? This is the conical structure. This is the egg. So here will be the sperm nucleus. Here will be the egg nucleus. This is the animal pole. This is the sperm nucleus. This is the egg nucleus. So these two will fuse here. Finally, to form a twice end nucleus containing both the chromosomes will form a twice end nucleus. This nucleus is known as synkaryon. This is synkaryon and this structure is the zygote. Finally, zygote is formed and the centrioles from the sperm, they come to the poles. So finally, the zygote is formed. One more thing, the part followed by the nucleus of the sperm will enter. So the part that is followed by the nucleus of sperm will decide the axis of cleavage of zygote. So in order to form the embryo, this zygote will again divide into various stages. We'll see in the next part. So that division, that starting of that division will be given by the nucleus of the sperm. Through the path through which it enters, from there this zygote will divide. Means that will decide the axis of cleavage of the zygote. Now these are the various steps of fertilization. Now we'll see what is the significance of fertilization. So one very important significance of fertilization is genetic variation. The sperm and egg chromosomes will fuse, so there will be genetic variation in the zygote. Next, meiosis 2. Start of meiosis 2 after fertilization. Then the egg retains cytoplasm. Due to meiosis 2, the ovum will retain all cytoplasm. Then metabolic activities also increase. Metabolic activities will increase. Lastly, the centriole of the sperm will give the first zygotic division. Okay, so this is the significance of fertilization. So this is over, fertilization is over. The next part will be the division of zygote. Means zygote is again divided into morula, blastula, gastrula, formation of 3 germ layers, and after that finally the embryo is formed, and after that we have parturition, then lactation. Okay, so after this we'll start with the division of this zygote.